Hi everyone, this is Ben speaking, and my teammates are David, Patrick, and Wilson. You can see them on your screen. Hey, please look at our PowerPoint. Yeah, our topic is dehydration. Remember this word because we will mention it later. By the way, in this presentation, we are mainly talking about the phenomena of dehydration on food, not on human body. Okay, before we start, let me ask you a question. Have you ever thought how people preserved food before refrigerators were invented? The answer is dehydration. See, I told you to remember, right? Food dehydration is one of the oldest operations used by the food processing industry. Here are our contents. I will elaborate the abstract of the experiment, and the rest of our teammates will go for the other ones. We have observed that some crops like potatoes and carrots would dehydrate due to salinity. By the way, salinity is the fat of containing salt. Thus, we conduct further experiments about restraining the growth of mold with the control of salinity. In order to perform the experiment, we used an ancient way to preserve food called pickling. Our purpose is to preserve slices of pickled apples and calculate the mold cover surface area. Furthermore, we could observe and discuss the features and the kinds of mold growing on the apple slices. Consequently, we have come up with a hypothesis. We think that apple would lose its water after pickling, which makes mold hard to grow. Now is the end of my part. Let's welcome Wilson for his pages.
Okay, let's talk about the procedures of the experiment. First, we cut one apple into slices, and all of them are in the same volume. Second, we sprinkle 5 grains of salt onto some slices, while the others don't. And then we place them into paper bowls and add 50 milliliters of water mold. Finally, we conduct 3 sets of the experiment. These are our variables in the experiment. First, these are our control variables, including surface area, the amount of water we add, and forms of paper bowls. Those are under control in order to make sure the experiment is conducted under the same condition. Next, this is our independent variable. We sprinkle salt on some apple slices in order to understand whether the salt is added or not will influence the growth of mold. Lastly, this is our dependent variable, growth situation of mold. And here are our materials we used in the experiment. We have water mold, apple slices, salt, and paper bowls. It's the end of my part. Next, Patrick will be here to talk about our experiment in detail.
Hello, this is Patrick speaking. After we place the slices well, we have observed these slices for statistics and kept a record. Something important is that We only took the pictures of certain slices because they had the most obvious changes. As we can see, there are no changes to both groups on day 1. However, some changes appeared on day 2. First, the salts that added on slices all penetrated in both groups. And strangely, there was a smell of fermentation in control group. Lastly, there are no obvious changes to both groups, which was similar to that on day 1. On day 3, some distinctions could be seen from now on. For the experimental group, they are still look same, but let's watch closely at the control group. We could discover that mold started growing. The experimental group still looked same on day 4, while the mold growing on slices of control group could be seen more clearly. By the way, water added in the control group nearly evaporated. In day 5, apparently, slices of the control group were covered with mold in large part. For the experimental group, we discovered that mold started growing on the edge of the slice. For day 6, the experimental group looks nearly all clear, while the mold that grew in control group occupied the surface of apple slices. That's all of my part. Let's welcome the next one speaking, David.
It's David speaking here. I'm going to draw the conclusion of the whole experiment. The chart shows the results of the experiment we performed. According to the experiment, the amount of mold we observed in the experimental group remained known in the first 4 days. In contrast, mold started growing on day 3 in the control group, and with a rapid growing speed, all of the slices in control group were all covered with mold on the last day we observed. Thus, as our hypothesis mentioned above, apple slices in the experimental group lost water after pickling, and mold would be hard to grow. And we also discovered that the kind of mold growing on apple slices was mostly the black mold. There are pictures of black mold, the kind of mold we discovered. Additionally, we found that we can only slow down the growth of mold instead of not letting the mold grow. Namely, the growth of mold is inevitable. Even if salt is added, mold will still grow, with the time being extended. Unlike the salted object in the market, they will not grow mold for a long time. We speculate that the water vapor in the open environment reduced the salinity, which caused the mold to fail to grow at the beginning, but in the end, it still grew. The site is where we got the information. Our presentation has finished and thanks for your attention.